Hello, my name is Maarten IJzerman, Professor in Cancer Health Service Research. Thank you for joining me today in watching this video where I will be talking about the health economics of molecular diagnostics in precision oncology. I will cover a couple of topics today. I first would like to start with some basics, the health economics of precision oncology, the use of companion diagnostics and how companion diagnostics really create value for money. Second, I'd like to talk about evidence development for new biomarkers which are not particularly related to drug treatment decisions or as a companion diagnostic. Thirdly, I will talk about diagnostic test efficiency. So I will show you some of the work that we do on real data analysis on molecular testing and how we can use this to estimate the budget impact of molecular testing, different molecular testing pathways, and whether there might be a case for complex genomic testing. And finally, I will show some of the work that we do on simulation modeling, which we use to fast track evidence development to inform new clinical trials in the area of precision oncology. So my first experiences with molecular diagnostics and particular liquid biopsies were way back in the Netherlands, where I used to work on circulatory tumor cells. So I used to work with a research group developing CTCs and the cell search technology, basically. Cell search is known for, it's been FDA approved and years ago. And we know CTCs, in particular the CTC count, and that has been shown in several papers since 2006, have shown to have prognostic value in predicting survival in different tumors. So this is, of course, in tumors where we are able to find CTCs, like breast cancer, colorectal cancer, prostate cancer. So if you take the arbitrary threshold of 5 CTCs per 7.5 mil, you will be able to identify those patients with a favorable outcome and a less favorable outcome. And this is quite convincing, and it's been shown several times. So at that time, the cell search being FDA approved, has been several trials, clinical trials have been performed, but there's no reimbursement. So that was kind of an interesting case for me to think about. So why isn't there a reimbursement if there's such a clear prognostic value of these CTC assays? So this is where we started to work on really using health economic methods to translate new technologies like CTCs into clinical applications. But there's more of those applications where we can demonstrate that's really challenging to bring new biomarkers into routine health services. So we all know the Oncotype DX, and this is just an example in Australia where the Oncotype DX was filed for reimbursement to MSAC way back in 2014, so it's 5-6 years ago, that it was submitted to obtain reimbursement, an MBS code to be able to use the Oncotype DX in breast cancer in Australia. And I have looked into the different meetings that ASK, the evaluation subcommittee, had, and MSAC. You see there's quite a few of those meetings, so ASK meetings and MSAC meetings, and up to June 2019, there was still no decision about the MBS code or MBS item number for the Oncotype DX. So it's a huge delay, 5-6 year delay in first time of submission and decision making. But here there's a delay, but there's also examples of technologies that are not even reimbursed at all. So this is the MammaPrint, which is a Dutch gene signature developed at the Netherlands Cancer Institute. And now it's quite similar to the MammaPrint, so there's quite a bit of clinical evidence, papers in New England Journal to aid decision making early stage prostate cancer, convincing clinical data here. And the National Health Insurance Board in the Netherlands in 2018 decided not to reimburse the MammaPrint in the basic package, in your insurance package. And the main argument for that decision is not about cost. It was about the risk of false negatives from lab tests. A fraction of the patients were having a false negative tests, which leads to undertreatment. They should have had chemotherapy, and have an increased risk of metastases and an increased mortality. So that was a reason for not reimbursing the MPCCC. So there's just 3 examples of biomarkers of molecular assays of tests that actually failed to get reimbursement. And this is just 3, there's probably many more of those tests. So as I said, it might have to do with cost. It's not always an issue of cost. So, and I would like to talk about the health economics here. And in particular, I want to make that point that health economics as a discipline is not about reducing costs. So that's not the argument, as in the MPCCC, this is not the argument of not reimbursing that the MPCCC test. In that case, it was a clinical argument, a judgment, a clinical judgment. We don't want to have the risk of a false negative. But health economics, important to remember, this is not about reducing costs. Health economics is about improving value and allocating resources in a way that we can maximize patient value. And health economists refer to that as opportunity costs. And opportunity cost means we can pay for one cycle of treatment with nivolumab, which is about $70,000 a year, 14 cycle treatment. But we can spend the same amount of resources to do 3 lobectomies, 3 surgical procedures. We operate 3 patients for the same cycle, the same course of nivolumab. And it goes even further. We can also do a 200 screens, CT-based screening, low-dose CT screening for lung cancer for this one course of treatment with nivolumab. So this is opportunity cost. The benefit forgone because we've spent all our resources in this one treatment with nivolumab. And the question for our health economists would be, what generates the most value for society? Would we spend it on the nivolumab, or would we better implement screening? Or do we do it all? That's the other question. But we assume there's a limited amount of resources, and we better make a fair judgment on how can we maximize value for society. So this is what we talk about in health economics, and health economists do a lot of modeling. We make the comparisons between treatments. So this is suppose with cancer X with 2 treatment options, A and B. This one is B is far more expensive than treatment A, and there's a difference in probability of success. But if there's a success of that treatment, you gain 2 life years. If you're on treatment B, which is the more expensive treatment, you have a 90% probability of success. You gain 2.5 life years. And here you can estimate the average life years gained from that strategy. If you would treat all patients with treatment A, you gain 1.8 life years at a cost of $20,000 per patient. And in the other treatment strategy, you gain 2.35. That's definitely more than 1.8 at the cost of 120,000 Australian dollars. So the question here is, I'm willing to pay an additional $100,000 per patient to gain 0.55 life years. So that's the question that we are addressing with health economics. Now let's move to companion diagnostics. That's on the other side here. We can still make the same decision, treat everyone with treatment A, treatment B, that's the 2 options, or we can decide to do a test first. We do a companion diagnostic test for EGFR, ALK mutations, and based on that specific test, we decide to start treatment. So if you're ALK positive, you would start with the treatment B. If you're ALK negative, you're starting with just chemotherapy. There's no targeted treatment available. That's the cheaper option here. So that's a very similar way. And if you do that test, that ALK test initially, you can demonstrate that on average, and I don't do the math here, but on average your life years gain is 2.34 at a cost of $60,000 per patient. So in this case, I can show you that you gain as much quality or life years with that test first strategy, 2.34 compared to 35, at about half of the cost. So that way, even though we pay an additional $1,000 for that single test, that single ALK mutation test, we can save a lot of money downstream. Well, we are not really changing the life expectancy or life changes. So that's where the biomarkers comes in, and of course then we have decisions if it's about biomarker development. So what's the minimum diagnostic performance? What's the diagnostic value, sensitivity, specificity, that specific biomarker to make that right decision? Like the case with the MPCCC, what's the false negatives and false negatives that we positives that we do accept given the decision that we have to make. So that's about the diagnostic performance. So that's in a nutshell what we do with health economics, health economic modeling, and campaign diagnostics. And we can demonstrate it in a different way. So suppose we've got the cost effectiveness plane. This is life years gained and the cost of the treatment. So we can plot a single treatment A, which is the chemotherapy, and that has a particular cost and a particular gain in terms of life years gained. So you can plot it on this curve. Then the second treatment that we're plotting on the curve is treatment B. So we treat all patients with that treatment B, which will be the ALK inhibitor, TKI inhibitor. So we treat all patients with treatment B. This is the life years gained. This is the additional cost. And you can show on your cost effectiveness plane here, if you would treat all your patients with that specific TKI inhibitor, this is on this side, this is way above the willingness to pay curve. Suppose this is the willingness to pay, which is $20,000 per life year gained. If we treat all patients with that treatment, that TKI inhibitor, your incremental cost effectiveness ratio will be far above the willing to pay threshold. So that means in this case, treating all patients would be way too expensive given what we want to pay from a societal perspective. But now if we would do a companion diagnostics, and let's go for immunotherapy, and we do PD-L1 expression for Pembro. If we'd only treat patients with PD-L1 expression more than 50%, this is the incremental life years gained, and this is the additional costs that are associated with that treatment, Pembro treatment, in that subgroup. And stratifying based on your companion diagnostic, based on your PD-L1 expression, means that you can really reduce your incremental cost effectiveness ratio. So in this case, it will be below the willingness to pay threshold, and that means society could make that decision to reimburse that pembrolizumab in case of PD-L1 expression more than 50%. So the case here, the willingness to pay, this is what society is willing to pay for that treatment, and this is actually what you have to pay for it. So that's the incremental cost effectiveness ratio. And in this case, your incremental cost effectiveness ratio is below the willingness to pay. So that means you can make a judgment to reimburse that specific treatment based on your companion diagnostics. So just a few examples of the value of companion diagnostics, and this is not just theory, this is real life, because this is several trials on PD-L1 expression based on the Checkmate trials and Keynote trials. And this is a nice one, the study from Switzerland comparing nivolumab to docetaxel, where you can see based on your PD-L1 expression, you see your incremental cost effectiveness ratio goes down. If you treat all patients with that nivolumab, your incremental cost would be $180,000 per life year gained. If you use that expression here, PD-L1 expression, and the threshold of more than 10% expression, then you can reduce your cost per quality to 126. So that's where you can demonstrate your companion diagnostic really has a value in terms of better tailoring the treatment, so to maximize outcome for that subgroup and to minimize cost of treatment for that subgroups. So there could be an argument to really get this reimbursed. So that's the companion diagnostics. So now let's move to other diagnostic, new biomarkers developed from larger studies to inform treatment decisions. And if people talk to me, they will say, well, we can demonstrate value of molecular diagnostics just based on just the budget impact calculations. And this is just an example I did. So suppose we've got a biomarker assay, which a target population about 10,000. The presence of that biomarker in the population is 20%. Cost of the assay is €100, and we can treat the patient. If the test is positive, it's 4,000. If the test is negative, we do another treatment, the non-targeted treatment, but we have a penalty if we start the wrong treatment. So if we start a wrong treatment, you have to continue with a second line treatment, which will cost you $2,000 in case of a false negative result. So you could do pretty simple math here. Suppose you have a test which is perfect. So with 100% sensitivity specificity, the total cost of this strategy will be 70 million euros. 70 million euros, and there's no waste because treating false positives. So treating a false positive means that they are negative, they should have had treatment, and you don't waste any resources here because you're treating false positives. Then the other test will be a no test, so that's random. So we treat at random, suggesting that 50% sensitivity specificity, the total cost of that treatment strategy will be 25 million. And in this case, you waste 4 million by treating the false positives, people that were positive from the test, but should not have had their treatment anyway. And this is more of an average test result, which would cost you total 20 million. So this is really a budget impact demonstration of what the value of a test is, the value of using no test at all, the use of a perfect test, and you can see there's a clear case, a business case, demonstrating the value of that diagnostic. Well, this is not just hypothetical, this is also real life, and I'd like to show you, this is the prostate cancer ARV7 test from Epic Sciences in Genomic Health. And this test is used as an assay to use CTCs to detect ARV7 expression on the CTC to determine resistance for anti-hormonal therapy in patients with metastatic castration-resistant prostate cancer. So that's a test. Test has obtained approval and reimbursement under Medicare in the US. And some of the work that's been done to support that claim comes from the literature here. This is a paper in 2016 where they did a similar budget impact estimation. We've got a number of patients eligible for treatment, 30,000 in the US, prevalence of the ARV7, so that's the percentage of patients resistant for anti-hormonal therapy, for enzalutamide and abiraterone, cost of the assay. So if we don't treat those 30%, we can save money. If we treat those 30%, we don't expect any gain, any benefit for them because they're resistant to that treatment. So we can save money downstream, about 20,000 per case per patient by not treating the 30%. And that means that the total cost, treatment cost avoided is 180 million, cost of testing the population is 30 million, so the net savings will be $150 million. So this is a budget impact study just looking at the cost of that test. Well, that's fine, but health economics is not just about cost. That's what I just said in one of the previous slides. We're not concerned with reducing costs. It's obviously very important to make a clear business case that you don't charge any more money to the system if it's not required. But I would be really interested in what's the additional value and additional cost. So if we develop a biomarker, if we develop a new assay, can we really improve patient value for a given cost? So let's look into, this is a little bit of research I've done in 3 tumor streams, melanoma, prostate, breast cancer, for different biomarker assays and tests that are being developed at the moment. And it's an impressive amount of tests for the 3 tumor streams. And they all are used in different parts of the treatment pathway, in screening, in early detection, certification, for tumor profiling, for decisions about aggressive treatment, more aggressive treatment, systemic treatments. So there's different ways you can use those tests. So I've did a bit of literature research on the prostate cancer tests, and there's quite a few using tests to avoid biopsy in prostate cancer based on molecular assays used in urine, so liquid biopsies. And there's tests that use the biopsy to genomic profile a tumor to determine appropriate systemic treatments. But let's look into those biomarkers. And this is just a bit of an overview of the different biomarkers with the company developing the biomarker and commercializing the biomarkers. There's quite a few of them. Some of them are also recommended in the NCCN guidelines. 3-4 of them are based on urine or on FFPE, and this is the decision they support. But I would like to talk about this piece here, the model or trial based. So you can of course look into the results, and it's all dominant, meaning that it's better to use a biomarker than not using the biomarker. I'll take that conclusion for granted. That's not what I would like to focus on. I would like to focus on model or trial. So all those studies, all published in the literature, are what we call model-based studies. So there's no prospective trial involved. This is just a model-based. They used a validation cohort, so they compared 100 of 200 patients did the molecular assay compare it to a gold standard, or try to figure out a relation between the presence of a specific biomarker and the prognosis of a patient based on a clinical validation study. But a clinical validation based on retrospective or cohort data is not a prospective validation of the study in terms of does it really improve outcomes. So those studies were model based, and my concern, they don't really provide the evidence that they do improve outcomes. And this is critical. In all the new biomarker development, we have to demonstrate the utility of new biomarkers. The utility to really change patient outcomes or to change patient management. So in this case, this is a paper where we presented the agreed checklist, which is about reporting economic evaluations of diagnostic tests and biomarkers. And 4 items are very important for that. So first, the first one that I think is important for us to consider today is that a biomarker, a specific test, has to be able to change clinical management. The test has to have an impact on the management strategy and the outcomes. That's the second. So you can change patient management. And second, you can change health outcomes and costs. So if you test really changes your pathway, really changes the decisions that you're making, so you can change the overall health outcomes and cost, that's the utility of a new biomarker. And the sixth one, or the third one in this slide, that is very, very important. What's the societal impact? So what's the wider societal impact of a chosen diagnostic and management strategy? So it's not just a patient outcome that should be better, but we also have to take that societal perspective, whether the implementation of a new biomarker assay has a societal value. We don't spend too much money from a societal perspective to generate a little bit of value for individual patients. So that's the pieces of work that I think are very important to consider in determining the clinical utility of biomarkers. Well, the problem is, if you look into the literature, there's not that many studies that prospectively evaluate biomarkers in a randomized controlled trials. I'm only aware of one trial in breast cancer, the SWOG 500 trial, and the other trial that I was involved in is the STIC trial. This is a French trial, from the Institut Curie. François Clément Bidard was the PI, and they actually used CTCs, a biomarker assay, to de-escalate treatment. So they had a randomization between 2 groups, a use your care strategy, a CTC guided strategy, randomized 2 groups, where they had an initial assessment based on clinical examination by the physician, with a decision to either start hormonotherapy or chemotherapy in the adjuvant setting. And so this is the initial assessment and the initial physician's choice that was made in both arms. Then they implemented the CTC assay and used a CTC count 5 cells per 7.5 ml to determine whether this is a high-risk patient or a low-risk patient. And if this is a low-risk patient here, so the physician decided to start with hormonotherapy, and the CTC confirmed this is a low-risk patient, the treatment started was hormonotherapy. But on the other hand here, if this is a low-risk or high-risk patient based on your CTC, where the physician has determined this is to be a hormonotherapy, this is the disconcordancy. So here we switch to hormone therapy. Well, sorry, in this case, this is a usual care. We don't switch because this is not disclosed. I have to be careful. In this case, we switch to the treatment. It's better to start here. In this case, this is the high-risk patients, and even though we initially thought we start with hormone therapy, because based on the high-risk profile of your CTCs, we decided to switch them to the CT arm, chemotherapy arm. The same can be done in the chemotherapy arm. This is concordant, and here there's discordance, so you de-escalate treatment from chemotherapy based on the low risk on your CTCs to hormone therapy. So the difference between the 2 arms, in this case, the CTCs are disclosed to the physician and they have to follow recommendations based on your CTC assay. In this case, these patients should have been switched based on your CTC, but this CTC information was not disclosed, so they were treated according to the physician's recommendation. That's the usual care arm. So this is the, I'll put it here, escalation group and de-escalation group. an escalation here and de-escalation based on your CTC assays. Well, if you do the trial and you look into overall survival and progression-free survival, this is kind of interesting. This is kind of no difference at all. So progression-free survival on this side and overall survival on the other side. Numbers of patients at risk in the trial followed for 42 months from time of conclusion. So that's no really a big deal in terms of progression-free survival. So you would say, this is a trial not to compare chemotherapy and homotherapy. This trial was designed to determine the value of that biomarker. So you would say here in this case, there is no additional value of using a CTC enumeration to guide that treatment decision. But that's probably too quick and dirty. So we looked into the different groups that were in the trial. So this is the concordant group, so the initial choice homotherapy and the CTC group homotherapy. And this is discordant, this is discordant, and this is concordant chemotherapy. So we looked into the different subgroups, so the homotherapy that were concordant and the chemotherapy concordant. There's no really a difference in terms of survival, progression-free survival, and overall survival. If you do an economic evaluation of that, you will end up here. So you add some cost to the system. This is on the y-axis, this is the cost. This is the x-axis with the live years gained. So this is a scatter of points here, which is all the patients simulated in the model. And on average, there's not really a significant increase in cost. There's no significant increase in survival. So it doesn't really make a clear case in that case. But now if we look into the other arms, and in this case, this is the escalating group. So here, initial judgment with homotherapy, and it ends up with chemotherapy. And in this case, we look in a second, chemotherapy switch to de-escalate to homotherapy. Here you could see a main difference. So if you switch to chemotherapy based on your biomarker assay, your life years really don't change too much on average. It's still, there's a huge variation around 0 gain, huge variation, but you only shift up the costs. So there's, you add cost to the system because you're implementing chemotherapy rather than homotherapy, which is more expensive. So you change cost in the system. It's going to be more expensive to use that chemotherapy or the CTCs to escalate treatment. But the final one, and I think that's the most important here, if you de-escalate, so if you use your CTC assay to be able to de-escalate treatment from chemotherapy to homotherapy, again, you won't change your survival outcome, because still there's a lot of variation in the survival gain, but you would change your cost. So you're reducing cost to the system because you're de-escalating treatment from chemotherapy to homotherapy. So this trial was not designed to demonstrate this value, but if you look at it from a health economic point of view, de-escalating treatment using your CTC assay would be the right way to go. So that's the results of that trial. It's not yet published, but the interesting discussion that we had last year with in France in Paris, well, the trial was designed 4 or 5 years ago, very nicely designed, prospective randomized controlled trial, one of the first in its kind to evaluate the biomarker. But at the time that we are able to publish the results, the treatment landscape has changed. So the decision here, or the conclusion was, if you look into all the new compounds that were introduced in that 4 years, palbociclib here was introduced, CD4, CD6 inhibitors, big 3 inhibitors were introduced for breast cancer, and they all changed the treatment landscape. So in that way, even though you have a very nicely designed randomized controlled trials, the treatment landscape is changing so fast that generating the evidence in a sophisticated, appropriate way would mean that you'd probably always be too late if it's about post hoc biomarker development. All right, the next topic here is of course understanding the implementation. So I've talked about companion diagnostics that were introduced in addition or as a co-dependent on the pharmaceutical component, PD-L1 expression to nivolumab or to pembrolizumab. We talked about biomarkers developed not associated with a specific drug treatment, just to de-escalate treatment, like the CTC assays. but the way this is implemented is really, really different between countries. So just a very quick overview. This is 2 countries, Australia and the Netherlands, where I've got a bit of expertise in. A very different way of making decisions about reimbursement. So in Australia, we've got a separate HTA process for diagnostics and services through MSAC and for drugs through PBACs. And this is listed on the PBS or in the MBS. In the Netherlands, there's a very standardized routine HTA process for drugs, for pharma, but not for diagnostics. There's no relation to diagnostics. So they assume this is going to be reimbursed anyway. There's no specific code, no specific process here. And if you look into the payment, and that's also very different. So the services here in Australia are paid. So you're paying for your companion diagnostics separately from the MBS. So there's MBS item numbers for specific molecular tests that are carried out. In the Netherlands, you just a bundle payment. So you pay a bundle, you pay a package of care, including your diagnostics, and what diagnostic procedures were done within the Netherlands is part of the hospital's decision, part of the clinical guidelines. There's no specific coding, no specific payment of that MBS. So those differences really make up the way new biomarkers are being reimbursed and what evidence requirements are required to negotiate reimbursement. So in case of Australia, say because there's an MBS item and you have to have an MBS item number to get a reimbursement for that companion diagnostic, we see that every new drug that's been supplied for reimbursement through the PBS with a companion diagnostic will have a separate procedure to get an MBS item number. And that ends up with a whole series of MBS item numbers for every single test. So we got PD-L1 expression, separate item number, and we got them for elk, for fish, and for Ross, and for EGFR. So we've got a cascade of different item numbers, and it creates a lot of bureaucracy because if you run a panel, you have to claim all the different tests separately based on the use of those tests for clinical decision making. There's no incentive, and it probably creates also a bit of inefficiency in terms of if you make that decision for every new drug which comes into the in the health system, in the PBS system, and every new drug will have a separate companion diagnostic. You end up with an array, a series of different tests that can be used and have a separate MBS item number. It might be much easier, much more efficient to claim a panel, an yes panel, to run all those mutations at the same time. That might be a much more efficient way to go through this. Anyway, this is where we are in Australia, and we can probably make a case that it's easier and more efficient to run those panels instead of sequential testing, cascade testing, like the reimbursement mechanism at the moment. And there's a bit of evidence, if you look into the literature, there is a bit of evidence that an NGS panel would be much more costly than running sequential testing. And the evidence mainly comes from the US. And this is a couple of quotes from different papers. You can have a look in those papers. NGS is expected to identify more patients with activation mutations. That makes sense. Thereby better enabling selection for targeted therapy and clinical trial enrollment. That's currently what we do in our phase 1 clinic too. But the budget impact, and that's where it's about, the budget impact to the US payers is expected to be minimally cost additive. So running a panel would create more opportunity to find the right treatments for the right patients, and the total budget impact, the cost implication is just minor. So that's a piece of evidence to run for panels, to go for panels rather than for sequential testing. On the basis of data from a nationwide oncology database, multi-gene panel testing has shown to be moderate cost effective compared to single testing in patients with lung cancer. That's a bit of evidence. So it's moderate cost effective to run the panel too from a large panel of patient database. And the final one is also about budget impact. Use of upfront NGS testing in patients with small cell lung cancer was associated with substantial cost savings and shorter time to results for both CMS and commercial payers. So that's also interesting. So running a panel it's not just cost effective or more or budgetly more efficient in terms of budget, your total budget. You can also probably improve patient outcomes because your flow, the workflow will be shorter, the delays will be shorter in terms of getting access to molecular tests and getting access to your treatment. So there's much more efficiency in terms of the workflow, the pathology testing workflow, and getting the patient the right treatment as soon as possible. So there's a couple of arguments to claim panels rather than single and sequential testing. But it's going to get a bit more complicated. So this is just an NGS panel, targeted gene panel for small non-small cell lung cancer for different tumor streams. But it's going to get a bit more complicated. So if you take, this is the picture I showed you before. We do treatments A and B. We can implement a campaign diagnostic to stratify patients, and we can do a bit of modeling, and we can look into the cost effectiveness of this strategy. That's all fine. But now we start using a comprehensive gene panel, and this is a paper from Annals Oncology 2019, where they run a gene panel with thousands of patients, several thousands of patients, where they were looking for activated mutations, and based on that mutations were selected for a specific treatment. So if you would analyze that in terms of your decision making and now what the treatments that were selected and the outcomes of the treatment would be much more complicated, and particularly if you consider the treatment associated with those decisions. And just to give a bit of an understanding of the complexity here, this is real world data that we got from several clinical registries in lung cancer, colorectal cancer, prostate cancer, with first line treatments, second line treatments, third line treatments, fourth line, fifth line treatments. And you can see the complexity of the treatment selection at the first line. So if you would have an activated mutation, a KRAS, what would be the optimal first line option in colorectal cancer patients, for instance? So there's so much complexity here. This is really personalized individual medicine. And if you now think about this health economic model that I showed you before, with we've got a simple decision to take here: do we test or don't we test? This simple decision, which is a binary outcome, yes test or no test, is emerging to a very complicated landscape with all kinds of mutations, with different actions, activated mutations, and different treatment options in the first and the second and the third line. So this is really individualized. And it's probably very difficult, not even probably impossible, to do proper health economic evaluation to inform that societal decisions. Should we allocate resources to pay for those panels, knowing that the complexity in the real world is this? So I'm just sharing the challenges in the modeling and the health economics, but also for HTA agencies, for regulatory agencies, making decisions that are beneficial for all of us in dealing with this complexity. So this is where we're heading towards. I'm not sure how that works out with health economics, but this is really the challenge. So just a little bit of a demonstration about the complexity in more detail. So this is a colorectal cancer data set where we had about 2,700 patients in the registry with different first line options, second line options, and third line options. And you see the numbers of patients receiving treatments in the first line. And what's surprising to us, it's only 16%. And we've got similar data for lung cancer, for prostate cancer, which is always also about 15 to 16% receives a third line treatment. So one of the questions that we've had in the research program that we're running is, why would you start with your best treatment to the third line? Why would you start? Because the window of the therapeutic window of opportunities is the most significant in the first line treatment. So why wouldn't we start with the best treatment in the first line here to improve patient benefit and survival? The other reason for demonstrating this is that we can use the real world data. So you can see a little bit of a simulation or visualization of that data here, but we can also use this to simulate different treatment strategies based on your biomarker assays. Okay, that's the treatment complexity. I'll come back to that simulation in a second. But the other complexity is the diagnostic treatment complexity. So this is real world data of the diagnostic pathway for lung cancer patients. This is based on Netherlands data, Dutch data, where we're able to mine all the patients in 2 years for the pathology testing that they underwent. Start here, imaging, lots of CT imaging is routinely implemented. Different panels used. And here you can see a patient flow in using those tests. So that's all real world data, that's fine. You can see the complexity again too. But we can use that also to figure out what's being tested at the end of the day. So what tests actually have been done sequentially or as a one batch in a cohort of lung cancer patients. So on the left here, you could see this is about 200 patients with all unique testing profiles. So the pattern of testing is all unique to those patients. So there's about 100 different combinations of tests that were performed in those tests just for lung cancer patients, non-small cell lung cancer patients. But based on those data, this real world data, we can do costing. We can allocate cost for a single test for a single mutation, and we can compute the total cost of the test for that individual patient. And that's what we've done on the right. So this is the total cost of the patient of the molecular testing for a single patient. So it's the range up to here 1,000 euros to 6,000 euros. So this is the total cost, the testing cost of a single patient. And this is the density, so the number of patients with that specific cost of the testing strategy. So you can map it out. You can map the total cost of the testing, but you can also compare it to whole genome sequencing. So in the previous example, I was talking about targeted gene panels, but now the question is, are we able to replace the standard of care testing with a whole genome sequencing? So we can look into this in terms of the percentage of patients in which there might be it might be economically viable to implement whole genome sequencing. So this is the current the total cost of whole genome sequencing in the Netherlands is about 4,700 euros for a whole genome sequencing. And you see the most of the vast majority of the patients have a total cost of testing which is far below the cost of whole genome sequencing. If you drop your prices, your whole genome sequencing price to, let's say, 2,000 euros, there might be a break even. About 50% of the patients might benefit from whole genome in terms of cost implications, and the other 50% might better go off with the standard of care testing. So that's just a financial and economic comparison. When would there be a trade-off? When would there be a break even for implementing whole genome sequencing from a cost perspective? So this is all based on real world data. Okay, so the final topic I'd like to talk about is the simulation. So what's the next step for us? So we can demonstrate, we can use with real world data, we can use real world data for treatment sequencing, we can use real world data for molecular testing, what tests are actually being performed in the real world, what's the total cost of the test, would it make sense to implement gene panels, targeted gene panels, whole genome sequencing. But the next topic and the next challenge for us is, if you know that complexity, which I showed you with the complex genomic testing, with the different activation mutations, with a range of different treatment options and different sequences, we know that this is going to be very difficult to generate the evidence, the clinical evidence for, in order to convince, to support claims to the MAPs, to the MSEC, to MBS about reimbursement. So that complexity, that's what I wanted to talk about. And what can we do? Because it doesn't make sense to design trials. Probably trials are too late anyway, as shown with the breast cancer study. But the complexity of this doesn't make it easy to design trials. So we have to think about different options. And one of the options is what we call simulation. And we've done a bit of simulation on circulating biomarkers for advanced prostate cancer. This is based on work that we've done with Johan de Bonen in London, in the UK, in metastatic castration-resistant prostate cancer. So that group of people that we worked with are all in support of economic evidence in addition to clinical trials and biomarker development studies. So that's all fine. That's the group we usually work with, and that's easy to convince that this is relevant. But the case that we start to work on is metastatic castration-resistant prostate cancer, whether we could use a molecular assay, a CTC-based assay, to inform about treatment progression. And if there's progression, we can use that to inform about treatment switches. So can we switch early on if there's non-response to specific treatment? And if there's non-response, what would be the best second-line treatment or third-line treatment that we can prescribe? So that's kind of the decision we would try to model. So it's really mapping a care pathway, the pathway of metastatic treatments. And this is some of the guidelines in metastatic castration-resistant prostate cancer. It's changing a little bit, obviously. First-line treatment, second-line treatment, third-line, and we've got real-world data to inform that treatment sequences. And you can compute the total cost of the treatment based on the three options here, starting with enzalutamide, abiraterone, and Kabasi at the third line. So that's the total cost of the treatment. So that's what you can do based on your current treatment pathway. Now the decision that we wanted to inform was, can we use an assay like CTCs to inform about treatment progression? And if there's progression, you would switch to a second-line treatment. So in this case, we start with docetaxel as a first-line treatment. We have routine monitoring of your CTC assays, and if there's an increase in your CTCs, that would inform us about progression. And there's evidence that your CTC assays outperform PSA as a biomarker of progression. So the case that we've made was that your CTC assay would be better able to detect progression early on. So that would mean that we can switch to second-line treatment earlier than with your PSA progression. So this is the pathway, the clinical pathway that we've modeled. So we start with the first-line treatment, docetaxel. We can make decisions all during the pathway to switch the treatment based on your follow-up to cabazitaxel in the second line. And there might be other options too. But that's basically the question. We follow a patient, we monitor patients, we make decisions to continue docetaxel or we switch to second line cabazitaxel. And then we can simulate the patients until they die. And this is basically what it looks like. I can put it on pause for a sec. Yep. Okay, this is the simulation model. So this is the start of the model. So we start with metastatic disease. We start with first-line treatment, docetaxel, monitor, and here this is a decision. At every time point, we monitor patients on PSA, bone scans, CTC. We can make that decision. Is there a response to treatment? So they are going back to the same loop. They go back to docetaxel. If there's not a response to that treatment, we make a decision to either switch to cabazitaxel after follow-up, or we discontinue treatment based on toxicity and other clinical features. The data, I mean, this is nice simulation. The data here comes from trials. This is not, this is real world data, but partly from registries on what we know about treatment sequences. But there's evidence from clinical trials, evidence about the efficacy of docetaxel as a third line treatment in this subgroup of patients, efficacy of cabazitaxel as a second line option. So that's coming from trials. So we synthesize different sources of information, different registries to inform the simulation model. And then we can run it. You can assume that as long as you're in treatment, docetaxel, you collect costs because you're on treatment. You have to routine hospital visits. We take blood samples. We do CT scans. We do PSAs. So as long as you're in the treatment pathway of docetaxel, you collect costs. You incur costs, and the same applies to the cabazitaxel. So that the total cost will be accumulated, and you can compute for every single patient that's flowing through this model until the end of the simulation, which is when they are dying. We can compute the total cost of that treatment strategy, and that's really running very nicely through this simulation. And this is what you will find from the run it again to go back to the final results. Yeah, sorry, this is the final results. So this is where, what I said, this is the total cost of your treatment strategy based on your CTC, because you can compare your CTC. Suppose you monitor them routinely, You can use your PSA, you can use bone scans, you can use circulating tumor cells assays as a biomarker to measure progression, and you can change your sensitivity, specificity of those biomarkers in order to inform that decision. So in this case, this is the total cost that you accumulate if you simulate those patients, 32,000 patients, using your CTCs. You can do the same for PSA, you can do the same for bone scan, and then you can compare the 3 different strategies. Which one would be better in terms of total cost of treatment? Which one would be better in terms of the total life years gained and quality-adjusted life years gained? So again, this is simulation, and this is what we try to use clinical trial data, real-world registries, clinical registries, administrative data to inform this kind of modeling. All right, so that brings me to the end of this presentation with summarizing the take-home messages for today. I started with the health economics of precision oncology with I think the conclusion that companion diagnostics, as I've shown you with the PD-L1, are a cheap way to create value for money. If you take the cost of PD-L1 compared to the total cost of the treatment, that makes a clear case. Then we moved to the development and the validation of new biomarkers, not specifically related as a companion diagnostic, for instance, the MammaPrint and the Oncotype DX. And I've shown you with the breast cancer, the STIC trial, that RCTs are the best way to generate that evidence on utility and cost effectiveness. But at the time they are completed and we can publish the results, the treatment landscape has changed. And this is not the first case. It will happen more often. I've shown you that we can look into diagnostic test efficiency, that we can make the case that complex genomic profiling is cheaper than sequential cascade testing. We can even demonstrate that at some point, if we can reduce the total cost of whole genome sequencing, we can make a case that implementing whole genome sequencing might be more efficient from an economic point of view, with the potential to drive much more, to generate much more actionable mutations to get the right treatment, patients to the right treatments early on. So I think the utility of whole genome or whole exome sequencing has to be developed yet, but in terms of the costing, we can demonstrate that lowering the price would mean that we can really compete to standard of care genomic testing. And finally, I've shown you with particular because designing trials with a long trial follow-up is a very inefficient way. I've demonstrated that simulation modeling using real-world data and clinical trial data might be a way to fast-track evidence development and informing new clinical trials or adaptive clinical trials. Thank you very much for your attention.